Welcome to the 6th lecture of Intro to Movement Neuroscience. As always, I'm very excited and have been anticipating seeing you all since last week. I hope that you thoroughly enjoyed last week's lecture and were able to review it a couple times and sort of take in the information and not feel too overwhelmed. Today, we're talking about the motor system, and we'll be finishing off with the tracks again. This lecture and the previous one, and I may have mentioned it last week, are my favorite to teach, just because it really starts to get into the systems neuroscience and is sort of that moment where we are piecing it all together. Was there any questions about the material from last week or anything from the asynchronous lecture? Okay, Eric has a question, everyone. My dad was reading Morse cause I'm sitting on it. Yep. Yeah, so I mean, if they were stuck in that position, if they died and gravity would like move their bodies, whatever position they're sort of held in, it would stick for a bit. And then it starts to like eventually things degenerate and then you lose the rigor mortis. Yeah. So Hannah asked a question, can we go over the M line and Z line stuff surrounding the muscle? So that is actually the muscle. So let me pull up the slide. Any particular question in regards to it, Hannah? Think of it like this is a cross, so this is like taking a zoom in of the muscle. It's like this, end on end on end on end in series the whole way. And so the active components of the muscle are right here. This is the myosin, and this is the actin filament. The myosin is what causes the cross bridging and pulls the two actin together. So contraction or the shortening of the muscle is as a result of all of these in series shortening. Shane has a question. In the GIF you shared, it looks like the actin keeps moving in one direction. However, it's moving. Yeah, it would be moving back and forth. Like this. Does that make better sense, Hannah? If you think of it like diagram, like think is it that's through the binding due to the actin being exposed then, like the contraction. Yeah, so if you looked at the let's see, there has to be a diagram here somewhere. So see how this is the actin. So this line is this. And in the presence of calcium comes in, the troponin tropomyosin moves off of the binding site for myosin off actin. So once that's gone, immediately myosin will bind and have its action of pulling the filament. And that's what happens here. So then it's like a cycle. It's a cross-bridge cycling because the myosin site's exposed, grabs, pulls, comes off, grabs, pulls off. Oh, there's Joseph. Okay, here we go. And the myosin were responsible for the movement. Yes, the myosin were responsible for the movement of those. It would be responsible for pulling on the actin. The actin is attached to the Z lines. So the Z lines or Z discs or whatever. It's just a way to describe where the actins bind to, or connected to each other. So it's all structural proteins. They called it the Z line because on a microscope it looked like a Z. But that's where all the actins connected. So then if they were to pull, it's the Z lines that get pulled together almost, right? So then think of this pieces of the myosin. See how it pulls here and here? It would pull these two things. Does that make sense, Joseph? Well, it's all really close proximity. So as long as the calcium site is there and removed the blockage from the tropomyosin on the actin site, then it will grab. It doesn't grab and miss. It's so close. It's the presence of ATP that will be whether or not there's enough energy for it to bind. Is H, so does it overlap actin or myosin? Hence the sarcomere has both nodes. Does it overlap actin? What does that mean? Like the Z lines. Oh no, so think about it this way. So look right here. So can we see the myosin, the red? See how when it grabs, it's pulling the actin closer to the middle. So if it pulls enough, this actin will overlap this actin. That's what it's talking about. So at full contraction, we have overlapping of the actin on top of each other. Does that answer your question, Tassi? Actin is the orange, yeah. Actin, myosin, the Z lines, it's all weighted to sort of describe the proteins, the structure of the muscle. Now, remember, this would be for per muscle fiber, it's that on N, on N, on N. So if we would go to like a diagram of a muscle fiber, you'll see like the striated pattern. There's got to be a picture of a muscle somewhere. See how it's like striated? That's because of the actin, myosin. So that's why it's also called, like skeletal muscle is called striated muscle, because it's striated like this. Does that make sense, everyone? For sure, everyone? And so a muscle fiber would be like hundreds of these on end. So all of the overlapping and so think if we had a whole contraction of the muscle, right? Globally, we see contraction of the muscle, but if we look microscopically, what's happening is that we have all these little bands shrinking at the same time. So if we get like eccentric contractions where you can break up some of the cross bridges, that is different. That's like the breaking of the cross bridges. Whereas with muscle tears, you're breaking fibers. So you would be ripping, like a rip would be through here. So it would, if you have like a strain or sprain injury of the muscle, you have like a torn muscle, you've torn through the muscle fibers globally. If you get a whole bunch of micro tears, it's a sign to the muscle, right? Because your body responds to the forces put upon it. So when you have muscle hypertrophy, it's your body's response to the little micro tears that are consistently happening because of your resistance training. So muscles will get stronger in one of probably 3 different ways. So you could improve your sort of coordination. You could change the firing rate or the number of motor units. You could also have muscle hypertrophy, so larger units. Yes, exactly. So with Joseph asking how do muscles get stronger, I think you mean like how do muscles be able to generate more force, like maximal force, so like bodybuilding, for example. because to have a stronger contraction, you could change the activation patterns, but you're talking more about how does the muscle itself get stronger, Joseph? Yes, because think about like the amount that you can, let's say if you wanted to bench press, right? Yeah, so if you wanted to bench press from like 135 to 225, part of that is coordination. So being able to contract your muscles in a way where it's more efficient. You can take one muscle and learn how to contract it so that muscle contraction of one specific muscle is stronger. Or the third part would be if a muscle itself is stronger and it has more like tension ability. So to say how do muscles get stronger, let me clarify again. Because either the way that you activate the muscles, all of them, the way you activate that one muscle, or the muscle itself getting a stronger ability to generate more tension. Yeah, so to get a muscle stronger or to be able to lift more weight, it could be one of those three ways. Like, I don't know if you guys have ever seen, think of like sometimes you'll see someone at the gym that's smaller, but they can lift way more weight than you expected. It's probably because their coordination and their efficiency in how to lift the particular weight in that exercise is better than someone that is like just huge and strong, but doesn't have the repetition or understanding of how to flex their muscles appropriately. Yeah, so when we say how does someone get stronger, I think that's more of a generalized term that could be a bunch of different factors. Whereas with what Joseph was saying initially, you can, when you say why is a muscle stronger, we can look into why a specific individual muscle could be stronger in terms of like hypertrophy. And that is the muscle's response to constant micro tears. The muscle will bulk up. So that doesn't happen. So when people say eccentric contractions are the way to generate more strength and hypertrophy, it's because you're holding the weight and all of the cross bridging that it's holding starts to rip. So you get little micro tears. Yeah, so then if you think about the larger the fibers, right? So they have a fiber versus a larger fiber, there's going to be more protein. So more of these, more cross bridging because there's more protein. Does that make sense? Yeah. So let's start with the new stuff. So based on that, because we're talking about muscle fibers here, so it's a good segue into the beginning of the synchronous lecture stuff. We all remember from the asynchronous lecture that a motor unit is a lower motor neuron and the specific fibers that it innervates. Does that make sense? So if we were going to contract a muscle, let's say there's 100 muscle fibers, let's say there's 100 motor units in a muscle, okay? If we wanted to contract it by 100%, you would contract 100 of the motor units, right? If you wanted to contract it by 50%, hypothetically, you would do 50 motor units. Each motor unit is innervated a certain number of muscle fibers. So there's more muscle fibers than there are motor units, okay? When we contract our motor units or we activate them, it's an all or none response that as soon as there's enough local potentials that the lower motor neuron will fire, all of the muscle fibers associated with that lower motor neuron will contract. Now, all of the fibers of a motor unit share the same characteristics, and that can be changed through training, but have we all heard of like slow twitch versus fast twitch muscles or muscle fibers? So given a single motor unit, all of those will have the same characteristics. So as you train from getting more, let's say you want to do like plyometrics, and you're going from, you want to like strengthen and improve the amount of fast twitch muscle fibers that you have. With the repetition of that type of contraction, your body is going to start transitioning from slow twitch, certain ones from slow twitch to fast twitch. So the force characteristics, or the different types of characteristics for a muscle fiber, can include twitch characteristics or speed of contraction, the force or tension ability, as well as fatigue ability. So speed, force, and endurance are the ways that we can classify different muscle fibers. And so we can classify motor units or muscle fibers. It sort of means the same things, because each of the muscle fibers within a motor unit will be of the same characteristics. And then as you train, you're training all of those motor units. Does that mean it's more efficient to train the muscle's native muscle type, like walking for bigger calves, rather than fast movements? Well, depending on the training that you're doing, you're going to be activating those muscles that are natively for that purpose, yeah. So if you see a big walker, they're going to have big calves. You see a basketball player that's like jumping, they have big gastroc, small calves, or thin. And the reason why that is because if you're training, you're activating a certain type of contraction that's going to be highlighting or maximally activating those fibers. And so with repetition, those fibers, the ones that are just like the fibers that are a little bit slower or like a little bit outside of that range, are going to start transitioning to that type of muscle fiber. I noticed endurance is one of the characteristics. Is muscle endurance referring to how long? Yes. So if you look at these, before initially people used to think it was just slow twitch versus fast twitch, and then they realized that it's sort of a spectrum, that you can sort of transition from slower twitch, medium, all the way up to fast twitch. And so it's a spectrum. But here on this diagram, we see this idea of a spectrum. So here we have one muscle fiber. It's fast twitch. It has high force, so we can see that it's per twitch, it's able to generate a lot of force. But in terms of fatigue, if we were to hold that contraction, see how it can only last for about 2 minutes. Whereas on the other end of the spectrum, this is a slow twitch fiber. Per twitch, it generates less than 10 grams of force. See, it's a lot weaker, but it can last for a lot longer. This is like up to 60 minutes. Eric's asking why you would want slower or fast twitch. Yeah, and exactly what Arvin's saying. It's depending on the task. So sometimes we're going to need endurance. Sometimes we're going to need power. And so you need both, right? Like we need a lot of our daily life stuff, like our postural muscles, they have to be slow twitch because it's something we have to do consistently over and over and over and over again. Whereas for fast twitch, they don't last as long, but they can generate significantly more amounts of force. So think of it like, so Shane's asking, does different lower motor neurons innervate fast versus slow twitch? Yes. So every motor unit has only one lower motor neuron associated with it, right? And so all of the muscle fibers of that motor unit are of the same characteristics. So there's going to be some that are natively fast twitch and some that are natively slow twitch, just like red meat and dark meat in like the chicken, if any of you eat chicken. But with training, you can generally start changing the muscle fibers of that motor unit into either, slower or faster. Yeah. So Carl's bringing up like a physiological perspective or exercise phys, where they're looking at the physiological reason why the muscles are either fast twitch or slow twitch. And it's because the slow twitch, they rely more on oxidative processes, whereas fast twitch, they're relying on muscle glycogen, which is glucose, sugar. So the image of the myofiber, which one, Shane? So the image of the myofibril. You could say the ribmen is fast and slow. No, you mean like so this is like fast, slow, fast, slow, fast? No. These would all be the same, right? These are all just a muscle fiber that runs the length of the muscle. The striated pattern is because of the proteins. So this striated pattern is in all of the skeletal muscles. This striated pattern, that's what the muscle fiber is. If you were to look at the muscle globally, wait, which one? If you were to look at, so if you were to zoom out and you would see all of these, it's going to be redder because there's more blood in it. There's more blood vessels because it would be for oxidative, with oxidative processes or slow twitch. Yeah, so don't think of it that there's like just a strictly slow twitch or just strictly fast twitch muscles. There's ones that are natively one versus another, like soleus versus gastroc. But the muscles are innervated by specific motor neurons. So if you want to train muscles, you are changing, by activating, you are changing the muscle fibers of that motor unit. So there are individual motor neurons for every muscle. A lower motor neuron innervates a certain number of muscle fibers. That doesn't change. Right? So then it's not like if I'm going to transition from slow twitch to fast twitch, I'm not changing connections, right? Let's say I want to have more fast twitch ability. I'm not taking my lower motor neuron and making it go to a different spot. I'm taking the lower motor neurons that are already there, and we're going to learn about motor recruitment. But what you're doing is you already have your lower motor neurons that are strategically fast twitch. You're going to start transitioning the other ones to have fast twitch characteristics. Yeah, you're not changing the connection. You are changing the composition of the muscle fibers. Yeah, perfect. Is everyone okay with that? Yeah. Perfect. So based again on the muscles, you can classify them based on their sort of motor unit characteristics. So what was that again? Speed, tension, endurance. And then with training, you can alter the amount of fast or slow twitch fibers in the muscle by having some motor units change their characteristics. So through training, you're changing the physiology of the muscle fibers with the constant repetition of whatever type of training you're doing. You're going to change the muscle fibers, and that's going to make a muscle fiber change to either slow twitch or fast twitch. And remember, it's not one or the other, it's a spectrum. And so like I was saying, if you want to contract a muscle, remember a motor unit is an all or none response. So once there's been enough stimulation to activate a lower motor neuron, all of the muscle fibers of that motor unit will contract. So the way to contract a muscle, let's say bicep, maximally 50% or by 10%, is by activating more or less motor units. Does that make sense? And so what we're going to talk about now is motor unit recruitment strategies. So to get a degradation, or sorry, to have a gradation of force, a gradient of how much force you can produce in the muscle, you're going to be recruiting more and more motor units. So let's talk about how to do that. So let's say we had our biceps muscle, and there was how many motor units for, let's say 100. 100 motor units in our biceps muscle. So that's hypothetical, okay? If I wanted to contract the bicep by 50%, how many motor units would I activate? 50, right? If I wanted to activate it maximally, how many? Yeah, okay. So that is motor recruitment. Some muscles, like the very fine ones in our hands, and think about our ocular motor muscles, they can only have like maybe potentially 10 motor units, or like 50 fibers. They're very, very small. So they don't have an ability to keep adding more and more motor units because there's already so little. So very quickly, if they start recruiting, they're at max of recruitment. So the other way to increase the force of a muscle is by rate coding or increased frequency of discharge. Because remember we're talking about cross-bridge cycling when talking about muscle, or the way that muscle contraction happens. You could fire the motor unit very quickly, and then you have a summation of the force. So those are the two ways to have a sort of gradation of force within a muscle. Oops. Yeah, exactly. Temporal summation. Right? So this is different than local potentials, but it's similar to like spatial versus temporal summation. You can add more and more motor units, and you can take the motor units that are there and fire them quickly. When talking about motor recruitment strategies, there is what's called Henneman's size principle. So have you all ever heard of that before? So Henneman's size principle, so Henneman was the person that, or the scientist that figured this out, but Henneman's size principle dictates that as we go and recruit more motor units to increase the force of a particular muscle, we go in order of increasing size. Slow twitch muscle fibers are very small. Fast twitch fibers are very big. So we sequentially increase the size of the motor units. So let's say there was 100 motor units in our biceps, like we said. We would start off with the slowest one, and then every time we recruit more and more motor units, we would sequentially go to the next biggest motor unit, the next biggest, the next biggest, the next biggest. So it's an orderly principle. Why would we do that? What would be the benefit of the sequential ordering of motor recruitment based on size? Yes, and so Manine and Aaron are both right. Because think of it this way. Which of the muscle fibers are fatigue resistant? Slow twitch, right? And what size are the slow twitch ones? Perfect, right? So you're going to start off with the slow ones, because if you're going to flex a muscle, you may have to keep contracting it for long. So it's in our best interest to activate the endurance ones first, right? And then work from that. The other thing was, because if every motor unit that you sequentially recruit is just a little bit stronger, it's a more efficient way to control force, so that every time you activate more and more of the muscle, it's a stepwise increase in force. If it wasn't that way, I did a small one, then a strong small one, big one, small one, big one. Like when I recruit more and more motor units, it'd be kind of a shaky movement, right? Because you wouldn't know the tension of the motor unit or the strength of the motor unit that you're recruiting. When you say recruit, are the motor units recyclable? Like after you use them once, can you use the same ones? No, yeah. It's just like a refractory period. The motor units, you always use it over and over and over again, which is why temporal summation can happen with, or rate coding can happen with motor equipment. Does that make sense? Wait, hold on. Time out. You guys are talking about recycling. So every time we flex the motor unit, we don't have to recycle. We don't have to break down the muscle. Yeah, exactly. Yeah. So does that make no? I read through back, Sean. So does that make sense, Selassie? Yeah. So okay, so think of it this way, right? You would get enough local potentials, right? If there's enough information coming down from your upper motor neurons, right? Based on the amount of activation, right? The amount of stimulus that's coming down, it's going to cause local potentials within the lower motor neuron, the dendrites of the lower motor neurons, right? The more activation will probably activate more and more lower motor neurons. But once the lower motor neuron has generated an action potential, it's going to go down, contract the muscles, and then there's the refractory period, and it can keep happening. So when we were talking about training, so remember this, Joseph. So there is, the motor units are placed in an order of increasing size. So with training, what you're doing is, if you're constantly going up to a point and activating the big ones, right? So you're going to be contracting the muscles enough to keep activating the big ones. The next ones in line that are, the next biggest, they're going to start training or changing to become bigger. If you were going to want to train endurance, right? If you're going to train endurance, so the muscles that are mostly natively fast twitch, like your gastroc, you're going to be holding those contractions. So the ones that are, you're holding the contraction, you're going to be contracting all of the fibers, and the big ones are going to die out because they're tired. So it's going to naturally want to decrease the size of the ones that are bigger to make them more endurance. Hopefully this doesn't make sense. Hopefully my hand movements really just demonstrated that for you. Does that make sense, everyone, in terms of like training and why training would sort of change the characteristics of a motor unit? Yes, exactly like that, Hannah. I would say that probably, yeah. Like it would be changing the composition of the muscle. So if you're looking at it globally, so your soleus is going to get bigger, your gastroc will probably get smaller. So globally, you won't really notice a difference. But if you were to take the muscle fiber, like take a cross section, like this is like exercise fitness, take the cross section out, you would see that the muscle fibers are decreasing. Because remember, with oxidative muscles or slow twitch, they require a lot of blood flow. So that's why they're red, and the fibers are smaller, but the muscle is potentially the same size in a sense because there's all these blood vessels through it. So in terms of bench though, if I did 1,000 reps, let's say they'd shrink more to be endurance. No. So resistance training is not about reps. So if you did 1,000 reps, you're going to get big muscles. It would be 1 rep and hold it. Yeah. Right. So so let's say you want to do 1,000 reps of bench press. That means you're going to need to develop more muscle glycogen to have sugar in there, so that way you can keep using those fast twitch. Exactly, Hannah. Yeah. Yep. So that stimulation is causing either a left or a right transition. Yes. Yes, Jessica. Yeah. Interesting, right? Oops. So here we see the difference in the size. But we all get the size principle, right? Because my research is looking at how Henneman's size principle with chronic pain, your body starts taking off the slow, like it changes the recruitment. It's in certain areas, it turns off the small fibers and jumps right to the big ones. You can all be participants in my study. So when we have, so this is do we all know what EMG is? So if you're looking here, so this is like this force output of a muscle. And this is the EMG of a recording of the muscle that we're contracting. Can you see how the EMG is larger for the larger contraction? So that would be because with increasing contraction, you're recruiting more motor units, and therefore that's more electrical potential. So for smaller muscles, and once all motor units are recruited, you can still increase the force of a muscle through changing or increasing motor unit firing frequency. This is temporal summation, just like Sean had said, and it's the similar process to the local potential temporal summation that we had talked about in the previous lectures. So when there is successive twitches of the same muscle, there's a temporal summation of the force. So if we look at this slide, if you have a, this is a wave summation. So see how if you generate a twitch right after, each one of these blips is an action potential. If the action potential starts after the force is already stopped, it kind of just is a wave. There's no real summation. If you have the firing frequency of the lower motor neuron at about 20 to 30 stimuli per second, the force in the muscle starts to add up, so we get temporal summation. It's not sort of like fully fused, but we do get a tetanus, meaning we have like a contraction, a sustained contraction. Whereas if we have a firing frequency of up to 80 to 100, that's when we see it. So see the blips. That would be the motor neuron firing 80 to 100 times a second. This all happens really, really fast. But then once you that amount of frequency, you get a fused tetanus. So you get a sustained contraction. So that term is called rate coding. So would fused tetanus be in slow muscle then? In all of them. It's just dictating the amount of like fires per second you need to get to a fused tetanus. Ultimately, if we're going to be using rate coding in our muscles, we want it to be a fused tetanus. Or else, like, your body doesn't want like jumpy muscles, right? This is just experimentally shown. That's why they labeled it. Exercise physiologists probably were like, okay, well, how many fires do we need a second to get a fused muscle contraction? So Shade, it is like that, but that's also because of like the muscle's tired. It could be pain. There could be a lot of reasons why it's shaky. But yes. So the more exhausted your muscles, the less fused the tetanus may become. Potentially, yes. But just like remember we were talking about training, there's a whole bunch of different factors. So I can't just say like, yes, that's the reason. Honor, what is the stimuli in voluntary muscle contraction? Yeah, what are you talking about the neurotransmitter? The stimuli would be your motor planning areas sort of activating the upper motor neurons to go to the lower motor neurons. Yep, that would be like a sustained isometric contractions, yeah. So when Shane was talking about like getting shaky, think about it more when like you're holding a muscle, like you're holding a sustained contraction and it starts to shake. So think of it this way, that so Shane's asking if wave is unsuccessful, is it like the hiccups? So remember how many muscle fibers are within a motor unit and how many motor units are within a muscle? They're not all firing at the same time. So it's like a cycling. So globally, it's going to look like a sustained contraction. So unfused, wave, fused tetanus is more looking at like a microscopic muscle fiber contraction and understanding how many fires per second you would need for it to be sustained. Yep. So whether it is through muscle contraction or through tension from pulling the muscle, the Golgi tendon organ is really just recognizing the tension. It's only responds to, like, receptors are a specific thing. It's only responding to tension. So based on the fact that now we understand that to increase the force of a muscle, you either recruit more motor units or you take the motor units that you have and then fire them. So you either do motor recruitment or rate coding. What we notice is that larger muscles rely more on motor recruitment for increasing force, whereas smaller muscles rely more on rate coding. And why would that be? Why would smaller muscles need to rely more on rate coding than motor recruitment to generate more force? Yeah, it's just this, it's the number of motor units. Yep, so you're all right. So here, like, so blue is, look at this graph here. Here we have the percent of maximal voluntary force production. So they're contracting the muscle from 0 to 100%. Okay. And this is motor recruitment. So this is activating 0 to 100% of the available motor units of a muscle. Smaller muscles in blue, when you get up to 100% of motor unit recruitment, sorry, see how it's only at 50% of the available force. The rest has to be rate coding. Whereas larger muscles, when you start individually recruiting the motor units, when you get up to 100%, you're already at like, what is that, 90% of force. So the last 10% is going to be rate coding. And that's strictly because there's more motor units in larger muscles. It's more EMG. So someone was saying they're going to do EMG in lab, probably in motor control, right? So this is EMG, using these two electrodes to sort of generate the electrical difference. And it's a way to register EMG or muscle activity. So it's used a lot in studies because you can record the electrical activity of the muscle of interest. And then you can sort of, what you do is whatever muscle you're studying, you get the participant to flex as much as they can. So that would be 100% of muscle activity. And then you do a study, and then you would record electrical activity as a percent of what max was. And then you can kind of suggest that you understand how much the muscle is firing by 0 to 100% based on this calculation. So that's how electrical activity from recording the electrical activity in the motor units is used in research. Does that make sense, everyone? Am I losing anyone? No. Is everyone okay? Awesome. Awesome. So now let's talk about reflexes, because that's also going to be contributing to movement. Okay? So do we all understand where we have the connection from our upper motor neurons to our lower motor neurons? Remember how we were talking about last day about the somatosensory system? How many neurons was it for our conscious relay pathways? How many neurons from receptor up to the brain, somatosensory area? 3. Okay. Motor neurons, or the motor system, it's 2. You go upper motor neuron, lower motor neuron. Okay, so so it does, but one second. Talassi, the 3 neuron, oh, what am I doing? I lost this. The 3 neuron pathway wasn't an interneuron in between. It was, let's say, the dorsal column. It was receptor up to the medulla, medulla crossing over to the thalamus, thalamus to the appropriate area of the somatosensory area. So none of those are counting the interneurons, meaning that an interneuron would be going back into smaller connections. So Hannah's asking, does that make more sense, Talassi? Yes, because it would be going in different areas. But then Hannah was asking a question. The synapsing in the spinal cord, you're talking about the motor neurons? Yeah. So then Joseph's asking, how does it go to the lower motor neuron then? Wouldn't synapse be lower motor muscle to muscle membrane? Yeah, so watch this. Okay, so all of you guys, come back up here. Think of this. Where's the picture? Right here. Okay, see here? So let's say this is our motor planning area, right? So we've decided that we want to activate whatever muscles, right? from our primary motor cortex. So that neuron starts here and travels all the way down through, all the way down to the appropriate part of the spinal cord, or sorry, the appropriate level of the spinal cord. And then it synapses with the lower motor neuron out to the muscle. And then with what Talasi was saying, well, doesn't it synapse from lower motor neuron to the muscle? Remember, it's a motor unit. So that's one functional unit. So if you activate a lower motor neuron, you are flexing the associated muscle fibers. So Joseph, does that make more sense? Yes, the upper motor neurons are in the brain and the brainstem. They're traveling through. And then they go all the way down to the appropriate level of the spinal cord. Yeah. So like wire one, so like right, and then out. So yeah, it could be the spinal cord, it could be the lower motor neuron, or what typically happens is if we have an upper neuromotor neuron lesion, like so let's say it was a problem with the upper motor neuron, it's not lack of reflex, we have clonus and we have like spasticity. And that would be the proximal axon. Proximal axon, what do you mean, Joseph? So neurons would still connect to interneurons, but interneuron, yes. Yeah, the interneurons are always going in multiple directions and all sort of all over the place. Think of interneurons as a way to, so signals are diverging or converging. I'm still waiting on Joseph's question about the proximal axon. Typically, proximal and distal axons. Okay, so then which part can I clarify for you? Show the paper. Do you want to go on the mic and ask the question?
Yeah, so like I remember one of the asyncs, there was something about like a proximal axon and like a distal axon. So like the distal axon would be like connected to the receptor. That's from like the sensory motor lecture.
So usually they use that for pseudo unipolar neurons, because technically it's one big axon with the cell body out to the side.
Okay. So the upper motor neurons then wouldn't have a proximal and distal, just be one? Then it just goes down all the way?
No, because so remember with sensory neurons, there's the dorsal root ganglion, right? So there is the receptor axon up to the dorsal root ganglion, right? And then the next that goes up to the medulla. When you're talking about the upper motor neurons, the cell body is right at when it begins. And then it's all just one axon. The rest is all axon.
Okay, I think that makes a little bit more sense.
For sure?
I think so.
So then with your sensory neurons, right? So think of like the dorsal column and the spinal thalamic tract. You have receptor, right? Where the receptors, and then you have the one axon that takes it up to the dorsal root ganglion, right? And then the next axon either goes to the spinal cord or all the way up the spinal cord to the medulla. Whereas with the motor neurons, the upper motor neurons, it's starting off in the motor planning areas. So there is like that, there's probably a little bit of an axon, but that's all just dendrites right there. The cell body is right at the very beginning, and the rest is all one axon. So proximal and distal is a way to describe the axon, and that was talking about specifically pseudo-unipolar neurons. Okay, perfect. As long as it makes sense, then, because chances are everyone has the same questions, right? What was Hannah asking? That would be because relaying info to the lower motor neurons causing contraction. And if there was an issue relaying this info, there would be an issue in the movement area. Sorry, that's confusing. 1 second, Hannah. Do you want to go in the mic and ask the question, Hannah? Those.
To be honest, it's not really relevant now. It was like when you were talking about like the stimulation of like a muscle and like the constant like stimulation of it. That was it.
Oh, so you're good?
Yeah, I'm good.
Okay, perfect. So let's talk about the lower motor neurons and sort of the synapses and connections that are involved in movement specifically. We've talked at great length about alpha gamma coactivation. So with telastine, we're talking about interneurons and how they sort of diverge or converge signals. There's interneurons right at when the upper motor neuron attaches to the lower motor neuron, right? The lower motor neuron is the alpha motor neuron. An interneuron is going to also at the same time connect to the gamma motor neurons, which is going specifically to the muscle spindles. So here's an example of the interneurons or a reflex involved in movement per se. The lower motor neurons are all sort of connected. So all of the motor neurons for a muscle, so all of the lower motor neurons for a particular muscle, they're all organized like sort of together. So let's say this is the upper limb. Here we would have like pec, bicep, pec minor, serratus anterior. They're all sort of coordinated and grouped together. So the upper motor neuron would come down and then activate the group of cell bodies or the lower motor neurons that are ultimately going out to a particular muscle. And you can see on this diagram that how, like, it's like, look at this big muscle here. There's roots that go both to 2 different spinal nerves. And then if you were going to look at this from the cross-section of the spinal cord, these are going to be arranged from the top down based on location. So we have proximal muscles are located more medially in the gray matter, whereas the cell bodies of our lower motor neurons going to distal muscles are located more laterally. Flexors are more posterior, and extensors are more anterior. And it's just the way the sort of mapping is in our spinal cord. So in terms of coordination, in terms of movement, remember how it just is so much processing required to activate our muscles in a specifically coordinated way that with spinal cord reflexes or the coordination that's provided through the spinal cord, it decreases the amount of processing required by the motor planning areas. One sort of simple reflex is reciprocal inhibition. So if I was going to flex my bicep, there's interneurons in the spinal cord that are going to turn off or inhibit the antagonist muscle. So if I wanted to flex my bicep, right? Everyone practice this. What's happening to your tricep? Is it flexing or is it not? Oh, she's asked a question. Yeah, purple is the lower motor neuron cell bodies. Yep. So it's relaxing, right? So the triceps relaxing if I'm flexing my bicep. Anytime we flex a muscle, we reflexively inhibit the antagonist, or else you would get a stretch reflex in the opposite muscle. And the reason why it's through the reflex is because if it wasn't that way, then my motor planning area is, okay, so let's say I decided I want to flex my bicep. Motor planning area figures it out, flex bicep. It would also have to say, okay, also at the same time, inhibit triceps for a particular action. It's like doubling the processing it has to require. So if it just says in this way, it's flex bicep, and then at the spinal cord, when it gets the flex bicep signal, it automatically, as a reflex, turns off triceps. Yes. When you flex your muscles, are you sensing the contraction of the muscle itself? You're actually sensing the muscle spindle contracting. You would be feeling the, well, because so proprioceptors are only give you proprioception. So length changes. So I'm sure that's like, you're going to be aware of that consciously. So that's going to be part of the sensation you're feeling. Sensation is how we are processing all of the receptors. So I believe it would be more of the sort of tension and like of the tissue. Yes, Hannah, exactly. Okay, so understand this, that the proprioceptive information that's coming from all of our muscles first has to go to the spinal cord, and then it goes from the spinal cord to the appropriate areas, right? That input into the spinal cord through interneurons can adjust and influence the activation of different muscles reflexively. And so what it's called is that at every level of the spinal cord, we have what's called a proprioceptive body schema. So the spinal cord is sort of aware in real time of the proprioceptors of the body, right? We know that that gets sent up to the cerebellum through one pathway, that gets sent up to our conscious somatosensory area through another pathway, but all of that proprioceptive information gets inputted into the spinal cord. And then what happens is that reflexively, if I, okay, so has anyone ever have you ever done this to someone where you're like, okay, hold your hand in front of your face, and then you get them to flex, and you let go, and then they hit themselves? Right. So your body will take the input from the proprioceptive information that's going into the spinal cord, and through interneurons that will reflexively change the activation intensity of different muscles in different ways. So by having muscle synergies or having proprioceptive input sort of alter the activation a little bit, it again decreases the processing required. The more reflexes we have, the more processing the brain that's already doing so much has to do. Here we have an example of autogenic inhibition. So, Joseph, remember you were talking about the Golgi tendon organs? Okay, so if the Golgi tendon organs fire, let's say there's an increased amount of tension. So the autogenic inhibition is a reflex that if there's too much tension in the muscle, it reflexively turns off. Now, anyone that works in a rehab center, have you guys ever heard of PNF stretching? So that's how this works. PNF stretching works by increasing tension in the muscle. If you increase tension in the muscle so much, the Golgi tendon organs are going to register that it's excessive force. It will reflexively turn off that muscle. So here we have so increased tension in the muscle. It's going to start firing the Golgi tendon organs, and that is going to go in, because it's sensory, into the spinal cord, and then reflexively through interneurons turn off that muscle. So see how its dotted line is inhibition? Do you all want to see a example? Here, one second. Hey, Kayla, can I borrow it for a second? I'm gonna give them a demonstration. Yep. You want to introduce yourself? They can see you, but you can't really see them. Everyone say hi.
Oh, it's so tiny.
That's
Hi,
not,
guys.
that's not what you said. Hey! Okay, lie on your back. So, I'm showing them piano stretching.
Ooh, okay.
Okay, so what I'm gonna do is I'm gonna show them. So this is like, okay. So Kayla works out like crazy. She's like on like a huge like training regime. Well, actually, it's not bad.
What's that one?
Like your hamstring
Oh.
flexibility. So this is her hamstring. Okay. Now what I'm gonna do, PNF stretching, is whatever muscle you want to stretch for the person, you get them to flex that muscle. So watch. So flex your hamstring into my hand. Harder, 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 harder. Now let go. Wait, not really flexing. Push. Push, 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 push. Now let go. Okay.
Ooh.
See? So do it again. Push, 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 push. Let go.
Oh my gosh,
Yeah.
really?
You can push. And let go. So every time she pushes as hard as she can, she is reflexively going to turn off that muscle a little bit
That's
based on
crazy.
autogenic inhibition.
That's so crazy.
Yeah.
Am I good?
Yeah.
I should do that myself.
And then, well, let me see, stay there. Actually, I should be doing it with you every day.
Yeah, you should. I still get treatment from him.
Try to find the reflex hammer. 1 second, everyone. Okay. So the myostatic reflex, the tendon tap reflex, is a little bit different. So can you guys see that? It's right there.
It's
What's right there? What's right there?
It's so
Can
involuntary.
you guys see that? So, any of you, what sensory process is responsible for this reflex? Sidebar, your spinal cord's okay.
Oh, is it?
Yeah,
That's really cool.
based on that. Woo! Oh yeah, no. So it is the muscle spindles that does this. I'll explain it. Okay, good. Thank you. Everyone say thank you to Kayla.
Bye.
So So autogenic inhibition is the GTO, whereas the myostatic reflex is the muscle spindle, right? So Golgi tendon organs, they register tension in the muscle. What do muscle spindles perceive? Length change and speed of length change, right? So the stretch reflex, if I am going to flex a muscle very quickly, it fights back. So when you tap someone's tendon, you are quickly pulling muscle fibers, probably close to the tendon, but at a high speed, it's very quickly. So reflexively, that gets sent back to your spinal cord, and the interneurons go right back and want you to activate that muscle. So autogenic inhibition is when you have too much tension in the muscle, it reflexively inhibits that same muscle. Whereas with the phasic stretch reflex, which is the myostatic reflex, two different terms for the same thing, it is the quick length change is getting you to flex that muscle. So it's activating the muscle. Does that make sense, everyone? So it's like the body trusting our reaction giving in with autogenic, but not trusting us with the spinal. Wait, say it again. So spindle. No. So the autogenic, so PNF stretching is taking you is sort of like a method to take advantage of a reflex that's already there. Autogenic inhibition is that if we are holding something that's too heavy, I'm holding it, it's too heavy, and it's so heavy it's gonna break. Like let's say I'm trying to catch something that's dropping, and it's so much tension it's gonna rip the muscle. The tendon registers that it's too much tension and turns off the muscle before it has a chance to rip the fibers. Yeah. So they're both safety mechanisms. Whereas if someone is so with the myostatic reflex, if someone's going to pull on you, you instinctively want to fight back. So they're both for safety. Because there's more to that. So now, I think, remember, Joseph, you were talking about the withdrawal reflex and cross-extension reflex? So here's another example of reflexes that will activate our motor neurons, but it's not through coordination in our upper areas. It's all through spinal cord reflexes. The withdrawal reflex is the sort of reflex that allows us to withdraw from a site of pain. So if I touch a hot pan or I step on a tack, that pain stimulus, before it gets sent up to or reaches our cerebrum, it's going to reflexively activate the flexor muscles of that limb to withdraw it from pain. So if we take a look at this example here, someone is stepping on a tack. What did Brandon say? So when we are stepping on a tack, that pain response is going to get sent up to the spinal cord and then right away activate the flexor muscles of that limb and inhibit the extensor muscle. So it's like withdrawing the limb from pain. And anytime we get a withdrawal reflex in the lower limb, we get a cross extension reflex on the opposite limb. Because the reason why this works is that if I stepped on a tack and then I lift up my foot, I have to plant the other foot or else I would fall, maybe onto the painful stimulus. So the cross extension reflex happens at the same time of the withdrawal reflex, whereas painful stimulus, let's say I'm stepping with the left foot, that painful stimulus gets sent up to the spinal cord. Inner neurons are going to send that reflexively, send that signal down. So we're going to flex or activate the flexor muscles of the left leg and inhibit the extensor muscles. So the left leg comes up. And then the cross extension reflex means that as soon as that happens, we're going to activate the extensor muscles of the right leg and then inhibit the flexor muscles of the right leg. Does that make sense, everyone? This is just a schematic for that. So this is all due to the lower motor neurons and the reflexes in the spinal cord. So when you're checking someone's reflexes, you're checking for whether or not you're checking for the integrity of the lower motor neurons and the spinal cord. If there's an upper motor neuron lesion, you get spasticity. Okay, is that because upper motor neurons would be conscious for sensing? Explain what you mean, yeah? Yeah, upper motor neuron would be for control because we're controlling it. So spasticity is a muscle that is sort of contracting and not letting go without conscious activation of it. Does that answer your question better, Hannah? So those reflexes are happening sort of all just in real time, just without conscious activation of them. Whereas the upper motor neurons, because they're coming from the cerebrum and in the brainstem, they're coming from the top down. It's just that in the brainstem, sometimes you can, like, you're not, I wouldn't say conscious versus unconscious, voluntary movement maybe, but it is like you're activating your core muscles from the premotor cortex. You're not consciously saying I'm going to activate these muscles. It kind of is like how it all gets planned. Yes. So then, yep. Yeah, so the reflexes are just an interaction between the lower motor neuron and interneurons in the spinal cord, whereas that stays like at that level, whereas the upper motor neurons are coming from above. Okay, perfect. So Shane's asking, so spasticity is uncontrolled but not reflexive. Like random depolarization or hyperpolarization. So actually, the reason why upper motor neuron lesions present as spasticity is because there tends to be, remember how with like neuromodulation and with local potential summating excitatory versus inhibitory, you're losing the baseline inhibition that comes from the upper motor neurons. So it's just that like that whole system. So when you activate the muscle, there's not a lot of like buffering. It's all just spastic. Because here, you know what, we're talking about spasticity. I'll talk about it in relation to the reticulospinal tract. Because we've talked about lower motor neurons thus far. Now let's talk about the upper motor neuron tracts. Like we talked about the somatosensory tracts last day, right? What are the three main categories? Yep. In the motor system, there's three groupings. There's the medial upper motor neuron tracts, the lateral upper motor neuron tracts, and then the anterior non-specific upper motor neuron tracts. Medial upper motor neuron tracts are for activating posture and girdle muscles. Lower motor neurons tracts are for fine control, and then the anterior non-specific motor neuron tracts are for neuromodulation of all the lower motor neurons in the spinal cord. So let's first talk about the medial upper motor neuron tracts. They're labeled that way, or they're called that way because they are medial. So if we're looking at the motor tracts, the lateral ones are lateral, the medial ones are medial. That's just why they're called that. And then the anterior ones are anterior. So when discussing the four different medial upper motor neuron tracts, they're all in relation to the activation of our core and posture muscles. The main one is the reticulospinal tract. So, Joseph is asking, what's the difference anatomically between the reticulospinal tract and the spinal reticular tract? Nothing. It is that, so reticulospinal goes from reticular formation to the spinal cord, the reticulospinal tract, whereas the spinal reticular tract is going from spinal cord up to the reticular formation. Yeah. So the reticular formation, right, is all of the grouping, is a grouping of the neurons in the brainstem. The reticulospinal tract is a motor tract, the upper motor neuron tract, that facilitates the activation of our core and posture muscles throughout our body. It's also, let me think of like, an example. So think of our activation of our core and posture muscles sometimes is unconscious, right? So if I am, okay, so you know when you are walking and then you slip on ice. If you slip or you start to lose balance, your body sort of readjusts very quickly. That's what's called an anticipatory postural adjustment. So evolutionarily, remember things that are happening in the brainstem, we're evolutionarily older. The activation of our core and posture muscles is evolutionarily older. It happens through this tract, the reticulospinal tract. No questions for a second. Okay, so that's one tract, the reticulospinal tract. The medial vestibulospinal tract is going from our vestibular system to the spinal cord. And what this does is it is all of the information from our vestibular complexes in our head to tell you which way the head is positioned. And it's a baseline activation of our head and shoulder muscles to keep our head stable in position. So depending on how my head's positioned, I'm going to reflexively activate the core muscles of the neck to hold my head stable. Whereas the lateral vestibulospinal tract, have you all ever heard of like someone falling asleep while they're standing? How can that be? Because if they're sleeping, they're not really controlling voluntarily their muscles. How come they're not falling down? It's because of this tract right here. The lateral vestibulospinal tract is going to register which way gravity is based on what our inner ear is telling us. And it's going to activate the extensor muscles of our body based on which direction gravity is. So medial vestibulospinal tract is giving our body a sense of which position our head is in and activating the appropriate neck and shoulder muscles to maintain its stability. Whereas the lateral vestibulospinal tract is registering which way is gravity and baseline activating our core and posture muscles based on which way gravity is located. Yeah, so classically think of it as this is all happening because the brain is already having to process so much that all of this can happen subconsciously. Okay? So the reticulospinal tract is the baseline activation of our core and posture muscles. Okay, so there's always going to be a baseline activation of our core and posture muscles, right? If we have too much, we get people that are rigid. Think about like when you know like zombies, when they show like zombies, like they're like, like when Walking Dead and all those different movies, how do they picture zombies? Spastic, right? Because in like zombies have just their brainstem. They're like the idea is that they don't have conscious control, right? So there's just activation of, I'm trying to make a joke here, but there's like activation of the reticulospinal tract of like the evolutionarily older stuff. if you know how if people have upper motor neuron lesions, like we were talking about the reflexes and spasticity before, it's because there's a baseline activation of their core and posture muscles without, when you have fine control, you're decreasing the postural controlling and allowing for fine control to come in. So without fine control, or the lateral upper motor neurons, you have baseline spasticity. And that's why you have it. Hopefully that made sense. Hopefully I'm not confusing you all. But zombies would be because of baseline activation of your core or posture muscles and no voluntary control of the fine. Talking about zombies. Is everyone okay? I know we're going over time, but as long as you're all okay, then I'm okay. I don't think my next patient's for another 20 minutes. Okay, so yeah, let's say it's fast zombies. Yeah, that doesn't make sense. Fast zombies has to be like an infection. It would have to be like increased levels of like being angry or something. Yeah. The medial corticospinal tract is for fine control of our core and posture muscles. So this is coming from the premotor cortex as well. So all of these tracts are subconscious except for the medial corticospinal tract, because cortico means cortex. So it's coming from cortex to spinal cord. Because I can still flex my abs, right? I can still consciously activate my core muscles. That's coming from this tract, the medial corticospinal, right? The other three, subconscious. Zombies can't do sit-ups, but they could probably plank. That's the spasticity. Is everyone okay with these 4 different tracks? Okay, perfect. This is the pathway. The lateral upper motor neuron tracts are for fine control, or what's called fractionated movement. Movement of muscles independently to each other allows for fine control. There's 2 tracts. There's the lateral corticospinal tract. So medial corticospinal tract from the last slide, conscious control of our postural muscles. The lateral corticospinal tract is for fine control of our fine muscles or the rest of our body. The lateral corticospinal tract is originating from the primary motor cortex. So Joseph, remember we were talking about upper motor neurons and how sort of it's situated? Can you see how now the lateral corticospinal tract is an upper motor neuron tract? See how it starts in the primary motor cortex and travels all the way down to the spinal cord, to whatever muscle that is. So what muscle would this would probably be? Leg, hip, I guess. So see how it goes all the way down. So there's not a proximal axon and then a distal axon. Cell body, axon. And then this would synapse with what? The lower motor neuron, and then go out to the muscle. So the lateral corticospinal tract, which is the largest, this is for fine control. It's in the lateral part of the spinal cord. These are all originating from the primary motor cortex. The rubrospinal tract is more for 4-legged animals. This is involved in facilitating the extensor muscles of the upper limbs, but we're not 4-legged. So evolutionarily, this isn't really a factor for us. And this is just a review of the primary motor cortex. Remember, there was the motor homunculus. Oh, sorry. Yep. That's true, Talasi. Yeah, just the functions, yep. Because remember, it's all about the wires and organization, just like the somatosensory area, just like the sensory system as well. You guys remember all of this, right? The different motor planning areas. So the motor planning areas are going to coordinate. So all of that from the frontal lobe, it's going to process, okay, what do we want to do? That's going to get sent to the motor planning areas. The motor planning areas are going to kind of figure out, okay, these are the muscles that we need to contract. And then that's going to get sent out from the premotor area down to the posture and core muscles. The primary motor cortex is going to activate the lateral corticospinal tract down to whatever muscles. And then we've talked about the supplementary motor area, which is going to coordinate series of movements or movements that involve two hands at once. And then Broca's area is for motor planning of the face when talking, the mouth. And then to bring it all home, remember we talked about neuromodulators and how neurotransmitters, if released into the sort of extracellular fluid, can float around and interact with like a large number of neurons. So we have our non-specific upper motor neuron tracts. There is the raphispinal and the cerebrospinal tract. Now they're named based on what nucleus they're coming from. But what this does is the raphispinal releases serotonin into the spinal cord, whereas the cerebrospinal tract releases epinephrine into the spinal cord. So it's a way to neuromodulate all of the lower motor neurons to be more likely to contract or not. Remember, serotonin was for arousal levels, epinephrine was for flight or flight. So once this happens, these tracks will cause baseline sort of neuromodulation of the spinal cord to affect how, sensitive, how hyperpolarized or depolarized all the lower motor neurons are. Does that make sense, everyone? Oh. Yeah, exactly. The rate's all hormones and how the nucleus and stuff, right? So probably when those hormones are released for digestion, it's going to activate this and then downregulate this. So that way, with digestion, you don't want the muscles to be ready to fight or flight. And so as like I promised, once you saw this diagram, we would get through the point where you would be able to sort of understand how the different tracks or how all of the wires of the spinal cord are put together. So now we've seen all of these sensory tracts last day, right? Dorsal column. This is the fasciculus gracilis and the fasciculus cunatus. So this is what? Fine touch, proprioception, vibration of the upper and lower body. Here we have the spinal thalamic tract. This is for pain and temperature. This is the spinal cerebellar tracts here. So that's going to the cerebellum from the proprioceptors and from the inner neurons. Here we have our motor neuron tracts. Here we have the anterior non-specific ones. Here we have our medial upper motor neuron tracts. And then this is the lateral corticospinal tract. This is for fine control. Was that okay, everyone? I know that was I felt a little bit all over the place, but I was really appreciative that you were all asking questions. I hope that I wanted to answer and highlight each one of your questions individually. I hope that didn't sort of break up the flow too much. How are we feeling about this lecture? Also, pyramidal tracts are up top, so where the medulla is crossing over. Ooh, so the final exam is actually, let me just see here. I got a word. It is, bear with me. Just pulling it up. Final exam is April 19th, 3:30. And next week is our midterm, okay? The breakdown of the midterm is very similar to the first one. It's 50 marks, 30 multiple choice, and 35 multiple choice and 15 marks for short answer. I think it's 3 or 4 short answer questions. For the short answer questions, you want to highlight the things that we've talked about numerous times. So on many different slides, we've talked about certain things. So Hera, I just can't come up. Like I've exhausted my supply of questions. Like I'm running out of like a number of questions to come up with. But for the short answer, know that it's going to be from what we talked about. I think it's going to be things that we talked about on like more than 4 or 5 slides. But also know that there's going to be a short answer questions, very small ones from the tutorial, like a case study. This concentration of ion will cause what? Yeah. I do, but I'm gonna email me because I'm recording this, so yeah. Yeah. You wouldn't, so any of the short answers that are from the tutorial, you didn't have to attend the tutorial to answer them. It's just like a case study type question that's taken from the tutorial. Yeah. No, I don't think there is. Previously, it was just to sort of give you an example of the type of questions that I would answer. Yes, exactly. Specifically similar to the assignment. But to do the assignment, you don't technically, you could have answered the questions without, like, just from the lecture slides, right? That's what I mean. Coolio? Everyone's good? Joseph, Hannah, Talassi, Carl, everyone's good? Okay, boom. I'm gonna go treat a patient. Bye, everyone. Remember, it's on Canvas, right? So the same thing as before, lockdown browser and monitor and all that stuff. Yeah. Okay. Bye, everyone.
Professor, I just had a quick question about so my situation with the course.
Yeah, I have no idea why. I don't know why they would have.
It turns out I've been like withdrawn from the course for some reason, so I'm gonna go get that sorted out. But I was just wondering if when I do get re-registered